Hi all, here's another 3-point test cross problem, problem 7.22 from the SNUSTAD textbook. Let's go ahead and look at how this problem sets up. We have a female who is homozygous for 3 X-linked mutations: yellow body, bar eyes, and vermilion eye color. So there's our original female. She's crossed to a wild-type male. That's Y plus, B plus, V plus over the Y chromosome. We can just sort of write it that way. That gives us an F1 generation, and the females in particular we're interested in. These are going to be heterozygous, so will look like that. And they tell us in the text that these F1 females have gray bodies, bar eyes, and dark red pigment. So the gray bodies and the dark red pigment, when we look back in the chapter, are the wild type. So effectively not the yellow mutation and not the vermilion mutation. Bar eye is a little bit trickier, and if you look at the end of the chapter material for chapter 7, Test Your Knowledge problem 3 discusses this a little bit, that basically for a female that has the heterozygous B plus B mutation, she would have bar eyes. The homozygous B or B plus B heterozygote for female would have bar eyes. The B plus B plus homozygous dominant would have what they called in the text kidney bean eyes. So some of the wording here is a little bit strange. It feels a little bit different. As well, representing the bar eye mutation as a capital B is a little bit different. But the results here are basically the same. We go on to say that this F1 is crossed by a male who is Y, B plus, V, and it yields the following results. Basically what we're looking at here is the same kind of idea. We want to identify what does the female's genetic orientation look like? What is the arrangement of her alleles? So just like we've done before, we'll look at our offspring. They've clustered these together for us, so we know that yellow, bar, and vermilion is one group and wild type is another group, gives us a large number of offspring. These are our parental types. And we know that all of the intermediate ones are the result of single crossover. And we know that the minimal number down here, the smallest number, are double crossovers. And so we can try to diagram this. First thing we need to do is figure out the gene order. And to do that, we'll take our parental chromosome arrangement. So this is yellow, bar, and vermilion, the 3 mutants. And the other one, yellow plus, bar plus, vermilion plus the 3 wild types. And we'll compare that to the double crossover types. Remember, it doesn't matter in which orientation we write these, but I'll write them just as they're written in the table: yellow, bar, and normal for vermilion. And the other one, normal yellow, normal bar, vermilion mutant. Again, we're looking for only one allele that changes between something written in the top row and something written in the bottom row. If it wasn't apparent in this case, we could switch the positions of our 2 double crossovers and sort of write them in the opposite places. But here we can see vermilion is the allele that changes in both cases. That tells us that vermilion is actually the gene in the middle. And so we go back and we rewrite this. Now that we know that, we can rewrite our F1 female as being yellow, vermilion, bar mutant, and yellow, vermilion, bar wild type. Again, we know the parental type, so we're all 3 mutants on one chromosome, all 3 wild type alleles on the other chromosome, and I've just rearranged it so that we're writing vermilion in the middle. Now to calculate our distances, we want to go ahead and look at what would happen here if we start with one chromosome and cross over in between the first pair of genes so that we can analyze that first intergenic space. So the single crossovers we're looking for here are going to be the ones that start with Y at the top, cross over, and give us V plus B plus, or the converse of that, starting at the bottom, Y plus, and then cross over for V and B mutant. We also have to remember that the double crossover types do have a crossing over event in this space, so we need to count them. That would be crossing over starting at the top, Y, crossing to the bottom to get V plus, crossing back to the top to get B mutant, and vice versa, starting at the bottom with Y plus, crossing up for V, and back down for B plus. And we'll count these up from our table. So our single crossover types, this one would be just yellow, and the one below it is vermilion and bar, so that's our first group. 244, they combine them for us, so that's nice. On our double crossovers, we know we're 50. Add those together, we get 294. If we do the same thing for the other side, now we're talking about starting at the top, crossing over, following that green line. Our single crossover types now are yellow, vermilion mutant at the top, crossing over to get bar wild type, or the bottom, Y plus, V plus, and then crossing up to get the B mutant. And then our double crossovers as well, the same as we wrote up above. Running out of room on this side, but we'll say here our yellow vermilion mutant, or bar mutant by itself, that's our second class of, there we go, color-coded. Our second class of single crossovers, 160. Our double crossovers were 50. Add those together, we get 210. And so what we can do now is that we can go and say, we know what our organization is, we know what our distances are, and so we can put it all together. We've got yellow, vermilion mutant, bar mutant, yellow wild type, vermilion wild type, bar wild type like this. We know our distances here, 294 out of 1,000 total if we add up all of those offspring, and then times 100 to make it a percentage. That gives us 29.4% recombination, or 29.4 centimorgans. The same math over here gives us 21 centimorgans. And so we know that the distance, 29.4 centimorgans between the left pair of genes, 21 centimorgans between the right pair. This is all this problem asked us. They just said calculate the distances, diagram the order, which we've done. I want to take this opportunity, though, to talk about the idea of coincidence and interference. We see that in a couple of these problems. And basically what coincidence is, or interference is the idea of, if we have a crossing over event happening in one space, how likely is it that we'll have a crossing over event in a neighboring space? And if you think about kind of all the machinery that must go into breaking the DNA and recombining it, moving the strands around and recombining it, you can imagine that that machinery probably takes up a little bit of space, like actual physical space along the DNA strand. And so you might further imagine that if two gene pairs are really close together, maybe if the crossing over machinery is happening in one spot, it's less likely that we'd have crossing over in a very close by spot. And that's what this is really about. So when we talk about coincidence, represented with the letter C, it's how often a double crossover occurs in practice compared to what we would expect. And so we'll see that that's represented as the number of observed double crossovers out of the number of expected double crossovers. And so In this case, obviously, the observed double crossovers we know was 50 organisms. The expected double crossovers, well, how do we know what that is? Well, that's the 1,000 organisms total that we counted, times the chance, or the decimal percentage, of getting a crossing over event in the first space. So this is our 29.4% that we saw in blue, represented here in a decimal form. And then we also have to have had a crossing over event in that second space. And so remember, anytime we use the and word, we're going to multiply in probability terms. And so when we do this, and we go ahead and we say our 1,000 offspring times 29% times 21%, we get on the bottom of this fraction 61.74. That means out of counting 1,000 offspring, we would have expected to see 61, 62 offspring that fit into the double crossover category. We only saw 50, and so when we do the math on this, we get a C-value of approximately 0.81. We could say 81% coincidence. Well, I, the measure of interference, is just basically what's left. So we say 1 minus C. So we take our 1 minus our 0.81, we'd say 0.19. There's 19% interference in these two pairs. So essentially what we're saying is the chance of a double crossover is 19% lower than we would have expected. This is problem 7.22. If you look around this section in the end of the chapter, 7.21, 7.23, there's several other examples that are similar to this. Some of them show you 4 classes of offspring like this one did. Some of them show you 8 classes of offspring where you have to identify the pairs. But remember, the parental types are always going to be the highest numbers that you see. The double crossover types will always be the lowest numbers that you see, or in some problems, the ones that you don't see, the ones that are so infrequent that they're actually zeros. And the single crossovers are in between. You need to figure out how they pair together based on your genetic diagram.